everyone. Thank you for joining us. It is a real pleasure for me to get to introduce you today to Dr. Mustapha Bouhrara. He's a SADMAN investigator and chief of the Magnetic Resonance Physics of Aging and Dementia Unit at the National Institute on Aging in Baltimore, Maryland. He works, among other things, with several large-scale studies. Most familiar to us is the Baltimore longitudinal study of aging, and he's incorporated additional brain imaging measures, brain structure, function, metabolism, and genetic variables to those studies. In my mind, he's particularly well known for his imaging of white matter, but of course he does way more than that. Today he's going to talk to us about progress in understanding, we changed your title, Progress in Understanding Cerebral Aging and Dementia using MR Physics and Neuroimaging Biomarkers. Please join me in a warm welcome, Dr. Bouhrara.
Thank you very much for the introduction, and thank you very much, Kristen, for the kind invitation and the honor to be among prominent names in the aging and also the neuroimaging field. So today I would like to discuss part of the work conducted in my unit, which is centered on understanding how changes in specific determinants of cerebral tissue contribute to aging and dementia. And for that, we combine MR physics with applied mathematics, neurobiology, and signal processing to introduce new imaging biomarkers or improve upon existing ones, including, for example, to map myelin content, axonal density, which will be the main focus of this talk. And the goal is to integrate them in internal cohorts, such as the BLSA and the Gestalt, which are internal cohorts run by the NIA, but also when it's possible, apply them to external cohorts. And I will show you some examples from the internal cohort, BLSA and Gestalt, and also from the ADNI, the DIAN, and the cardiac study. And the goal is to combine them with other imaging biomarkers from PET imaging and fluid biomarkers to have a more holistic understanding of the mechanisms of aging and dementia. So why imaging and why MRI imaging? So as you certainly know here, that Alzheimer's disease is pathophysiologically defined by amyloid plaques and tau aggregation leading to neural injury and then brain atrophy. And also this paradigm shaped much of our understanding of Alzheimer's disease. It's also kind of restricted our perspectives, because if we look at the bigger picture, it's clearly telling us that Alzheimer's disease does not only involve amyloid and tau aggregations, but it's a complex disorder involving many interconnected processes from mitochondrial dysfunction, iron accumulation, oxidative stress, demyelination, vascular dysfunction, neuroinflammation, axonal degeneration, and so on. And I believe to advance the field of Alzheimer's disease, we need to understand the specific implication of those mechanisms in the disease progression and also how they interact with each other, and also how they interact with the classical view of Alzheimer's pathophysiology. But for sake of time, today I will focus my talk on demyelination and axonal degeneration, often seen as downstream aspects of Alzheimer's pathology, but our research kind of suggestion that they may play a much more central role. So for that, I will start with a quick background motivating our investigations. And then I will spend a few minutes, or 12 or 15 minutes, to discuss how we can quantify myelin content and axonal density without going into technical details here. And then how we integrate those biomarkers to derive new insights about the implication of demyelination, axonal degeneration, and normative aging, and also across the clinical ID spectrum of consumer. So this work is motivated by mounting evidence from histology, transcriptomic studies, showing the early white matter degeneration and dysregulation of oligodendrocyte genes that are tied to myelin synthesis, even in the preclinical stage of Alzheimer's disease. We have also a study from Dr. Klaus Nav lab in Germany showing that demyelination might precede or even trigger amyloid accumulation to some neuroinflammatory aspects, kind of putting demyelination as an upstream factor of Alzheimer progression. But of course, these are mouse studies, engineer studies with their own bias. But for me, my personal interest is really on human studies, specifically on neuroimaging studies. And we have evidence from, for example, Dr. Barbara Bindlin from the Wisconsin-Madison, and I believe she is talking next month. Fantastic. And she showed that there is an association between lower myelin content and higher amyloid pathology as measured from the CSF, even if in asymptomatic individual that they were enriched for the APOE4, which is the strongest genetic risk factor for Alzheimer's disease. We have also evidence from Dr. Adam Brickman from Columbia University in a series of work, including in autosomal dominant Alzheimer's disease, or AD-AD. And the AD-AD population, it's a population with very rare mutations in the APP, PSEN1, and PSEN2 genes. And these populations were unfortunately developed the Alzheimer's syndrome at younger ages and overexpression of amyloid pathology. But the interesting aspect about this population is that they will develop the syndrome around the same age of their affected parent, allowing us to estimate, which is called the estimated year to onset, to symptoms onset. And then we can start looking at them at very early, or look at the brain changes at very early ages, and also at younger ages when all, I would say, age-related vascular issues are not present and can found in our results. So what Dr. Brickman shows here is that in the mutation carriers, they start to exhibit significant increase in white matter hyperintensities 2 decades before the symptoms begin. And the white matter hyperintensities, as you know, are those bright lesions in the brain that can be seen in structural imaging, and there is an ongoing debate about their etiology. Are they due to risk factors, vascular risk factors? Are they due to some endogenous aspects or genetic aspects, or white matter degeneration? So we are adding to this picture of white matter degeneration, even including in the AD-AD cohort, by showing that, but at the microstructural level, so using our biomarker that I will shortly talk about it, or the axonal density index, or ADI, that quantify axonal density, we can see that axonal density start to decrease in the carriers of those mutations around the same time, even in the normal appearing white matter. So we are far from those lesions. So this evidence, or those studies, kind of forms our I would say, a basis for our investigations to address 2 simple questions. Is white matter degeneration a hallmark of Alzheimer's disease? And if so, what exactly does it involve? Is it demyelination, axonal degeneration, or both? And the specificity question is very important, not only for the temporal dynamic of those changes, but also to inform specific and targeted interventions. But to address this 2 simple questions, I would say, we need to measure myelin content and axonal density. And to my knowledge, the only way to do that in vivo and longitudinally without killing the participants, of course, is through use of MR physics and imaging. So without going into technical details, cerebral tissue, it's a complex media. For example, in white matter, we have, for example, 3 different water compartments. We have the water surface between the myelin bilayers, we have the intraaxonal water, and we have the extraaxonal water. And MRI, it's mostly a water imaging technique, right? But the interesting aspect is that this water compartments exhibit different NMR properties, or nuclear magnetic resonance properties, such as relaxation times T1 and T2, which are simply the time for this water compartment to restitute the energy given to them during excitation inside the magnet. So in my group, we take advantage when we use multi-component relaxometry and take the advantage of the fact that the myelin tropics between the myelin bilayer exhibit different NMR, in this case, relaxation times, and we try to isolate the fraction of the myelin water fraction to derive this matrix myelin water fraction, which is a direct measure of myelin content. We also use the fact that the intraaxonal water and extraaxonal water exhibit different diffusion coefficient, and we try to isolate the fraction of the intraaxonal water and derive a parameter called axonal density index, which is a direct measure of axonal density. But unfortunately, quantitative MRI, it's inherently slow, especially if you want to acquire with high spatial resolution, full brain coverage, and also you need to scan fast because we are dealing with older participants or participants with cognitive impairment that cannot stay in the magnet for a long time. For relaxometry to estimate this little parameter here, the myelin water fraction, we can use which are called steady state imaging or SS imaging. So without going into technical details here, after many failures and trials, I will say we were able to derive using the block equations, which is a mathematical framework that allows us to track the magnetization over time. We were able to derive the correct analytic expression of those steady state sequences under this multi-component system to be able to map this little parameter here, the myelin water fraction. As you can see here, the problem with steady state imaging is very sensitive to noise, making the inverse problem. The inverse problem means that you go from the images to the parameter maps using this equation, very unstable. So you need to acquire the signal over and over many times to reduce the noise in these images and improve the stability on the inverse problem. Decreasing the scan time is possible, but it comes at the expense of increase instability of the inverse problem and reliability of the parameter estimation, in this case specifically is myelin water fraction. So to improve the stability of this the inverse problem, we simply adapted the Bayesian theorem, and this is one of the foundational, I would say, mathematical framework that has been around, I believe, over 2 centuries and a half now, and kind of shaped our reasoning and uncertainty has been applied across all scientific fields. So we simply adapted the Bayesian theorem to our equations, and we were able to introduce a method called BMC or Bayesian Monte Carlo analysis, allowing us now to have full brain coverage, high resolution, myelin water fraction maps within 21 minutes instead of 105 minutes that look fairly similar to the ones derived using high SNR, signal to noise ratio. We recently have been invited to write a review papers on the application of myelin water fraction imaging to study aging and neurodegenerative diseases. We have also another book chapter coming soon that is more focused on the technical aspects, and I believe those 2 documents could give a overall appreciation about the application of myelin water fraction imaging for those who are really interested. So in my group, we keep pushing the limits of MR physics to reduce the acquisition time without compromising accuracy. In a recent work conducted by by Zhao Yangong, Dr. Zhao Yangong from my group, we combined BMC with neural networks to introduce a new method called reuse, allowing us now to have full brain coverage, high resolution myelin water fraction maps within 7 minutes instead of 21 minutes. And you can see the pattern of demyelination in this AD subject were captured using this reuse analysis. But the big question, or I would say the 600-pound gorilla in the room, is how specific is our method to myelin content? And the direct way, or the most common way to validate the specificity of your biomarkers is through use of histology. But histology is very destructive. You lose water during the preparation. Also, the sample can be denaturated. So it's not really the gold standard, I would say, for the validation of those biomarkers, at least in NMR. So in my group, we've been thinking about how can we find new ways to validate the specificity of our biomarkers, and it occurred to us that we can leverage the power of omics, especially transcriptomics and proteomics. So in a work conducted by Dr. Jong-Yun Bae from my group, he's a postdoc in my group, what we did is that we correlated the myelin water fraction values derived from different brain regions. Every data point here, it's a brain region, that are expressing different amounts of myelin after aggregating the data from different participants in the BLSA and in the Gestalt studies, which are our internal cohort. And we correlate this with transcription factors, especially those involved in myelination, such as the MBP or the myelin basic protein, which is a canonical protein involved in myelination. And the MBP or these transcription factors were obtained from the Allen Human Brain Atlas after aggregating the data across all donors. And as you can see, there is a very strong and significant correlation between our measure of myelin water fraction and MBP expression. So then, in collaboration with Dr. Keenan Walker, who is an expert in neurodegeneration and proteomics, we correlated the myelin water fraction from the BLSA and Gestalt, who have also plasma samples. And we assayed that using the SOMA scan. We had like over 7,000 proteins, and we correlate that with the BLSA and Gestalt. I think it's over 100 and something participants here. And as you can see, the most strong associations and significance association were with proteins that were involved in white matter maintenance, especially oligodendrocyte maintenance and metabolism. So this omics, transcriptomics, and proteomics analysis not only provide a validation of the specificity of our biomarkers to myelin content, but I believe that it offers MR physicists around the world new strategies to validate the specificity of their imaging biomarkers using omics without having to deal with problematic histology. So for axonal density, what we try to do is to isolate the intra-axonal water because it's proportional to axonal density. There are two techniques that are well established. SMI, unfortunately, requires massive amount of data to be acquired, so it's not really clinically applicable, not at the time being. But there is a technique called NODI that is very applicable and gains a lot of momentum and has been integrated in many clinical investigations that can isolate this fraction and calculate, which is called the axonal density index, which is the measure of axonal density. So in 2018, I believe 19, I came across this and I was interested, and I implemented in our ongoing BLSA longitudinal studies and Gestalt. But I rapidly realized, and other groups, by the way, also, that NODI has some, I would say, issues. First of all, as you can see here, the CSF fraction, which is a parameter to be estimated as well, in the deep white matter regions can go up to 20 or 30%. And we know that this is not physiologically plausible. Those values in the deep white matter region should be close to zero. And also, as you can see from this map, this is a healthy young participants. I believe this is my brain. I suppose I'm healthy. Let's not see that I'm healthy. And you can see that the IDI values, even in the deep white matter region, can go to 80 or 90%. And this is physiologically plausible from our understanding from microscopy, postmortem investigations, and histology. So two methods have been suggested to address some of the issues that I discussed, but both of them come at the expense of extended acquisition time, and we don't have the luxury for that in our investigations. So without going into technical details, what we did here is we mathematically reformulated the NODI signal model, and we introduced a new method called CNODI, or constraint NODI. And what we did very briefly is that the CSF fraction of the isotropically diffusion water, it's a known parameter to be estimated in NODI. But in CNODI, we can estimate this parameter from structural images that are available to us in every clinical protocol, and then use it as a known parameter in the inverse problem, reducing the dimensionality of the parameter space, and then increasing the stability of the inverse problem. Again, the inverse problem is the mathematical framework that take us from the acquired weighted images to the parameter images, in this case, the ADI that we're interested on using our mathematical or biophysical model. So as you can see, the CSF fraction are close to zero in the white matter regions, as they should be. And also the ADI values are within the expected physiological range, around 50 or 60%, again, in these healthy participants. But the good aspect of it is all of this comes without extending the acquisition time, which means for us that we can use existing multi-shell diffusion data in existing cohorts, and multi-shell diffusion data are now acquired almost in all external cohorts. So to do like a larger investigation, and I will shortly show you some examples. So then to validate the specificity of those methods, or ADI, we correlated the ADI values derived using NODI or our method CNODI with NFL, which is a plasma biomarker of axonal degeneration. And as you can see here, ADI derived using NODI poorly correlate with NFL, but those derived using CNODI exhibit strong and significant correlation with NFL, with, as expected, higher levels of NFL or higher axonal degeneration or neuronal degeneration corresponding to lower axonal density. So here again, this proteomic analysis validate the higher sensitivity and specificity of our biomarker to measure axonal density. But as I said before, it also offers MR physicists around the world new ways to validate the specificity of their imaging biomarkers by using omics without having to deal with problematic histology. Just to summarize this first part of the talk, we developed methods to quantify myelin content and axonal density. Now the question is, how can we use these imaging biomarkers to address our two simple questions? Just to remind you, is white matter degeneration a hallmark of Alzheimer's disease? And if so, what exactly does it involve? Is it demyelination? Is it axonal degeneration, or both of them? So in other words, what we are dealing with as a scientific question is, are demyelination and axonal degeneration associated with cognitive and motor dysfunctions? And also, are they involved in differential levels in the AD continuum from the preclinical stage to the prodromal stage to the symptomatic phase? So to address these questions, we implemented myelin water fraction imaging in the BLSA and in the Gestalt studies, and we look at the association between baseline myelin water fraction and retrospective changes in cognition, including memory, processing speed, executive function, fluency, and attention. So Dr. Jiao Yan Gong from my group and Dr. Murad Bilgil from the Laboratory of Behavioral Neuroscience at the NIA use linear mixed effect models. And what they show is that participants with lower myelin water fraction or lower myelin content at baseline exhibit steep, as indicated by the pink line here, exhibit steeper decline in cognition, especially processing speed and executive function, and to some extent with memory. Then we look at motor function, and we look at the association between baseline myelin water fraction and retrospective changes in gait speed. And gait speed is a functional measure reflecting overall physiological performance and motor function. Here again, as you can see, participants with lower myelin water fraction at baseline, as indicated by this pink, I guess, line, exhibit steeper decline in gait speed. In other words, steeper decline in motor function. I just would like to emphasize that this is a very conservative design. What I mean by that is these are cognitively normal participants, overall healthier than the general population, and yet we still see the implication of demyelination in cognitive changes and motor decline. So given this association of demyelination and cognitive and functional decline in normative aging, we wanted to look at this association across the Alzheimer's disease continuum. Unfortunately, BLSA and Gestalt are mostly normative aging cohorts, so we cannot do that. But we were thinking of ways to leverage existing data from appropriate cohorts, such as the ADNI cohort. The issue here is that those cohorts do not include myelin water fraction imaging, or at least not yet. But there is some recent literature showing that the extra diffusivity or the extracellular diffusivity, especially the perpendicular to the axons, can be very sensitive to myelin changes. As you can imagine, when the axons demyelinate, the water in the extracellular space, especially perpendicular to the axon, becomes much free to move and increase the extracellular diffusivity. But the problem with this is that this framework was mostly a theoretical framework and also based on simulation. So we had to validate it first using in vivo data. And what we did here, we implemented multi-shell diffusion to estimate the extracellular diffusivities, perpendicular and global general. And we correlate that with myelin water fraction measures in BLSA and Gestalt. And as you can see, the extracellular diffusivities exhibit strong and significant correlation with myelin water fraction, with, as expected, lower myelin water fraction or lower myelination or higher demyelination is associated with higher extracellular diffusivity. So given this as this reassurance, I will say about the high sensitivity of these biomarkers, extracellular diffusivities, that can be derived from multi-shell diffusion that are available almost in all external cohorts. We gained access or secured access to the ADNI cohort, multi-shell diffusion, to derive the extracellular diffusivity perpendicular and global, and also the longitudinal cognitive and functional measures, in this case, the CDR-SB, which, as you know, is the clinical dementia rating sum of boxes, which is a measure of cognitive and functional abilities and dementia risk. So using, again, here linear mixed effect model, we look at the prognosis values of those of these biomarkers in cognitively normal participants CN and cognitively impaired participants CI. And CI, they were mostly like 90% of them were MCI. We are really looking at the really early stages of clinical Alzheimer continuum. So as you can see here, participants with lower DE, as indicated by the green line, which means higher myelin content at baseline, showed a slower incline in the CDR-SB, or in other words, slower decline in cognition and function. And interestingly also, this association was even stronger in this prodromal phase of Alzheimer's disease. And here again, participants with lower diffusivity or extracellular diffusivity, in other words, higher myelin content, exhibit a slower incline in CDR-SB, or in other words, a slower decline in cognition and function. And this is interesting because this myelin content, myelination in general, is measured using these parameters or myelin water fraction, may serve as an indicator of brain maintenance, which must be linked to some way, as explained by Dr. Yakov-Stern, to cognitive resilience and cognitive reserve.
Can I just ask, what is the beta referring to? Is that the beta for the interaction term?
Yes,
that's the time by 4-time interaction term.
Yes.
Okay, thank you.
So in collaboration with Dr. Galdo Alvarez and Dr. Rivas Fernandez from Spain, we using myelin water fraction imaging this time, we showed that demyelination is also involved even in subjective cognitive decline, and this is adding to our previous investigations of the implication of demyelination early in the AD continuum using myelin water fraction imaging, although we had very low N here, like a sample size. So we are currently investigating whether amyloid or tau pathology are involved in the demyelination, or whether demyelination is a completely independent process. And this will be interested not only for the mechanistic understanding of the interaction of those pathologies, but also for informing targeted interventions. So for axonal density, here again, we use the ADNI data, and we secure access to longitudinal multi-shell diffusion data, CSF data, or CSF biomarkers of AD pathology, and PET biomarkers of AD pathology. Again, here the cohort included cognitively normal participants and MCI participants. So using linear mixed effect regression analysis, Zhao from my group showed several things here. First of all, you can see that the CI group, as indicated by this orange line here, exhibits significantly lower axonal density at baseline as compared to the CN group, and also a steeper decline over time in axonal density as compared to the CN group. And this is consistent with accelerated axonal degeneration in MCI. So as you can infer from these graphs here, the effect size and significance of these findings were stronger with ADI derived using CNOD as compared to NOD or SMI. But another very surprising finding also was that none of the CSF or PET biomarker demonstrated a clear group differences in trajectories. And this is interesting because it points to the limitations of these biomarkers for disease monitoring, like a diffusion MRI, which can monitor the disease very, very sensitively. So then we examined the relationship between baseline ADI and future changes in cognition and functions, as measured using the MMSE or CDR-SB. Just to clarify, and you certainly know this here, the MMSE is the mini mental state examination, with the higher values means good cognition, lower values mean bad cognition. CDR-SB, as I said before, it's a rating scale to measure cognitive and functional abilities, but here higher scores mean bad, lower scores means good. So as expected, the CN group maintained a stable cognition and function of scores over time, but the CI group exhibit progressive cognitive and functional decline over time, as reflected by decrease in MMSE or increase in CDR-SB. But what is more interesting is that these cognitive and functional changes were modulated by the baseline ADI. What I mean by that is that participants with higher axonal density at baseline exhibit, as indicated by the green lines here, exhibit slower decline in MMSE and slower incline in CDR-SB, or in other words, slower decline in cognition and function. And again, this is interesting because it shows that axonal health, and specifically axonal density here, may serve as an indicator of brain maintenance and likely cognitive resilience and cognitive result. What's this slide? Oh, yeah. So here, just to show that the association was captured using CNOD and NOD and SMI completely failed to capture this association. So then we restricted the sample size to participants with both CSF and diffusion MRI, multi-shell diffusion MRI, or PET biomarkers and diffusion MRI. And as you can see, ADI derived using CNOD demonstrate predictive performance comparable or even better at some instances as compared to the CSF and PET. And here I'm showing just the best performing biomarkers from CSF and PET. But beyond this high performance or predictive power of ADI or diffusion MRI, we have to keep in mind that diffusion MRI have other advantages. It is not invasive, it's not radiotoxic. We can repeat the measurements as much as we want, and also it provides topographical information for detailed spatial investigations. So then we look at the DIAN study, or the AD, as I said before, the autosomal dominant Alzheimer's disease. And as I said, the DIAN cohort include this population of a very rare genetic mutation in the APP, PSEN1 or PSEN2. The mutation carriers were simply develop the dementia and also overexpression of amyloid beta. And the interesting part is that they were developed around the same age of their affected parents, allowing us to look at very early at the brain changes that may occur very early before the symptoms begin, and also at younger ages when all those age vascular issues are not present and confound in our interpretations. The DIAN cohort includes carriers, mutation carriers, and also their siblings, non-carrier siblings in blue. So the carriers are red and then non-carriers in blue. So previous work have shown that amyloid accumulation starts 2 decades before the symptoms onset, and this is something that we can confirm from our access to this data. But what is more interesting is that we also picked up signs of axonal degeneration showing up around the same time, especially if you look at using CNOD. And obviously here the sample size is limited, the confidence intervals are wide and ugly, but this is the same thing when we restrict the sample size for the amyloid cohort to the same number of participants. So then we push our investigation a little further, because in a previous work from the BLS and Gestalt, this is cognitively normal participants, we showed that ADI increases until middle age, around like 42 years of age, and then starts to decline afterwards. So when we did the same quadratic modeling to the estimated year to onset symptom, which is simply age, as we did before here for age, what we saw is that both carriers and non-carriers have this similar inverted U shape trends. But what is interesting also is that the mutation carriers, or the peak of axonal maturation in the mutation carriers, happen almost 3 decades before the symptoms begin and start to decline very fast as compared to non-carriers 3 decades before the symptoms begin. And this is, again, it clearly shows the implication of white matter degeneration, in this case, axonal degeneration, very early in Alzheimer's disease, even in the genetic form of Alzheimer's disease, and not only amyloid. So this project is ongoing because we are now moving to validate these ideas in another genetic form of Alzheimer's disease. I'm talking here about the Down syndrome. Sadly, most of them will develop early dementia and early amyloid accumulation, which make them a crucial population for testing these ideas. And another interesting aspect with the Down syndrome is that they virtually do not develop hypertension. So if I might say, we have a much cleaner population without those confounding vascular risk factors. So we just gained access to the ABC Down syndrome data, and we are very grateful to Dr. Adam Brickman for helping us and guiding us to get access to this data, and we're about to validate our finding that we found in the AD code. So in conclusions of this part is the finding that I showed from the ADNI, BSA, Gestalt, and Diane clearly showed that axonal degeneration is linked to cognitive and functional impairment in the preclinical stage and also in the prodromal stage of the clinical Alzheimer continuum, and also support the potential of ADI derived using CNOD as a biomarker to monitor the disease progression, but also for prognosis and phenotyping and in clinical research. Just to summarize the second part of my talk, we developed MRI biomarkers to quantify myelin content and axonal density. These biomarkers allow us to track the disease, to predict the disease, and also to understand the disease. And the big picture that we are trying to, or the idea that we are trying to work, or if those results, like our investigations are conclusive, is that it seems like the axonal degeneration and demyelination are early and central features of Alzheimer's disease and could represent new ways for interventions. Of course, amyloid and tau matter, but protecting also the brain wiring may represent a powerful way or additional way for prevention and also for treatment. So in this final part of my talk, I would like to briefly touch on risk factors, especially modifiable ones, metabolic and vascular, that may impact myelination and axonal density. And this is relevant because addressing these risk factors can prevent the dementia risk by 40%, according to the recent Lancet report from 2024. I think it's the updated version. The first report was in 2020, and the 2024 was the updated version. So in a series of work by my former fantastic students and my current amazing students, we show that obesity and hypertension are both associated with lower myelination. In an exciting work by Angelique from my group, she also found out that higher levels of HDL, which is known as the good cholesterol, was associated with higher myelination. And this is interesting because these factors, or these metabolic factors, are modifiable. It can be modified through lifestyle changes, exercise, or diet, or also through established interventions. But another area that we are aggressively pushing forward in my group is the vascular risk factors. And also here we showed that arterial stiffness, as measured using pulse wave velocity, was associated with the G ratio. And G ratio is an aggregate measure reflecting reduced myelination and axonal density, with higher values means lower myelin and lower axonal density. We also showed that, I just would like to emphasize, this is the MBSA and Gestalt. This is a cognitively normal participant overall here, and yet we still see those associations. So we also showed that lower cerebral blood flow, as measured using arterial spin labeling, were associated with lower myelin content in VLSA, and also in the cardiac study, which is an epidemiological longitudinal study looking at different vascular risk factors that can impact the brain. We also showed that lower cerebral blood flow at baseline was associated with steeper decline in white matter integrity, as measured using diffusion tensor imaging, specifically here is functional isotropy, for those who are familiar with this parameter. And this is interesting because cerebral blood flow, as an end product of all those metabolic and vascular risk factors, can be modified. It can be improved through exercise or also through use of phosphodiesterase inhibitors. For those who have no idea what I just said, this is essentially a Viagra. And there are some mounting evidence showing that Viagra can improve cerebral blood flow. And we are looking at this with our clinical director to design a clinical trial, because our hypothesis is that this association between improved cerebral blood flow through Viagra or phosphodiesterase inhibitors and cognition may be moderated or mediated by improvement in myelination and axonal density. My group also is interested in 6 differences in myelin and axonal density. In a recent work just recently submitted by Noam from my group, we found that sleep disturbances were also associated with lower myelin content, especially in women. Also, Nathan from my group showed that insulin resistance in women is associated with lower myelin content. So it seems that women's myelination is vulnerable to these risk factors. And in fact, in an ongoing work, we just start to scratch on this. We found that women have higher myelin content at younger ages, but it starts to decline much faster as compared to men, especially around the menopause. And we suspect that this has to do with the estrogen or the drop in estrogen level due to the menopause or the premenopause. And one of the key aspects of estrogen is that it helps transport the glucose to the brains, especially the oligodendrocyte cells to make myelin. And there is some evidence from mice models showing that in the absence of estrogen, the brain may turn in a ketogenic state and start using lipids, especially myelin, as a fuel. So we are working with our clinical director, Dr. Josephine Egan's, to measure the hormonal levels and finalize our analysis. And if it's conclusive, this clinical data may support our hypothesis and also provide new perspective on estrogen therapy given the disproportionate Alzheimer's prevalence in women. So I just would like to finish this talk with a note of hope and a positive note. So in the BLSA, this is a work that was conducted by my former student, Mari Faulkner. She's now in the Indiana University doing her MD PhD. What we found in the BLSA in Gestalt is that participants with higher cardiorespiratory fitness, as measured using VO2 max, also exhibit higher myelin content. What is also interesting is that this association was even stronger in middle age and older ages. And this is kind of suggests to us that lifelong physical activity may have some lasting benefit for brain health and especially on myelination. And why this is a positive or a note of hope, because there is no pro athletes here. There is no Michael Phelps or Serena Williams. These are like everyday individuals like you and me who maintain a, say, moderate exercise routine, and yet we can see the drastic impact on myelination. And of course, this is a cross-sectional study with its own limitations, but we are looking at this longitudinally in the BLS in Gestalt as well, acquiring longitudinal data. And we recently also secured access to the cardiac study, and we're going to look at the cardiorespiratory fitness versus white matter generation overall as measured using diffusion tensor imaging. So yes, several modifiable metabolic risk factors are influence myelination, axonal density. This is exciting because it opens the opportunity for clinical trials, and we are already engaged on that in the NIA and also with our partners outside the NIH. In conclusion, we showed that myelin, demyelination, axonal degeneration are likely key aspects in aging and also to higher extent in Alzheimer's disease. Of course, our models are biophysical models that are based on some assumptions and approximation, but this is exciting for the new generation to take over this development and improve the sensitivity and specificity of this imaging biomarker, which is always the case for any biomarker, including non-MRI biomarkers. There is always this need to improve sensitivity and specificity. The goal is to foster I would say, integrative program that combines MR physics with signal processing, mathematics, and neurobiology to develop or improve upon existing biomarkers for disease monitoring, mechanistic insights, characterization of the disease, phenotyping, and prognosis. So they say it takes a whole village to raise a child. I say it takes a whole institute and beyond to build a research group. And I'm deeply grateful to my former students, my current students, my mentors, my collaborators, and also the NIA and the NIH leadership for the guidance and also the support. And I would like to emphasize my special thanks to Dr. Zhao Yanggong, who is co-leading with me the MRPAD unit for the fantastic work that he's conducting. But my sincerest thanks go to the participants. Some of them have left us. May God bless their souls. Thank you very much.
Thank you. Thanks for not making us feel dumb for not understanding the physics either. That was a really good balance. Questions for Dr. Bouhrara?
I could kick off. So what do you think neurobiologically is the mechanism by which very early, take the Diane study, what is it that that mutation is doing that's leading to demyelination?
That's
22 years before symptoms.
Yeah, that's a very good question. I think that these mutations were studied mostly, and was expected also, and amyloid expression. but there is little evidence to link those to white matter degeneration. One of the aspects that we would like to look at is to run some GWAS analysis and also some Mendelian randomization analysis to see if those genes also are linked to white matter degeneration. The problem is that the sample size here is very limited, and as you know, for those GWAS type analysis and Mendelian randomization, you need like the genetic information and also the MRI information for at least 5 or 10,000 participants to have some reliable investigation. As we suspect that those genes, it's like the IPV4. IPV4 is the strongest, as you know, gene risk factor for Alzheimer's disease and amyloid pathology, but it also has different roles. For example, it improves cerebral blood flow, and also it helps in transporting the glucose to the cells, and glucose is the main constituent of myelin. And we have some evidence linking that, like the IPV allele 4 to lower myelin content. So I think that most of the interest was done on amyloid pathology, or I would say Alzheimer's pathology, but little has been done to link those genetic risk factors to white matter degeneration as an endogenous aspect.
Just a quick follow-up. Do you think that those findings would generalize to sporadic Alzheimer's?
Yeah, that's a very good question. So in sporadic Alzheimer's, which I show from the diagram, we see that axonal degeneration, demyelination are involved in the preclinical stage, and also to higher extent to in the prodromal phase, especially here I showed mostly from MCI, which is like a very early stage of Alzheimer's disease. I think we have to look at in sporadic, late onset Alzheimer's disease, also early Alzheimer's disease, and try to find ways to translate the observations that we are making from the genetic or familial or genetic, like at the Down syndrome aspect of Alzheimer's disease. Look at, for example, at regional distribution, which regions in the brain are more sensitive to those changes, compare them also to the sporadic Alzheimer's disease or late onset Alzheimer's disease to do some inference or translate. But we can also then do some genetic analysis in the sporadic Alzheimer's disease. The issue is just the sample size. We are not there yet.
I had a similar comment that if you could back the Diane group up even further to like childhood, you'd probably see super low intercepts on those measures too, right?
Absolutely.
Because it's genetic.
That's a very good point, because we are working with Dr. Yakel Quinrose in the Columbia cohort, Columbia, the country, not Columbia University.
Yeah.
We created our myelin imaging there, and we are looking at the offspring of people that, and we're also interested in sex differences. And we're looking at the offspring of those participants with those genetic mutations, because, as you know, they have like a large prevalence in Medellin in this region. And we will see. So I'm just participating in a grants for her studies, and we start acquiring the data. Hopefully, we can derive some insight to see if those changes might occur really earlier.
So your sequences will be in that new cohort?
They are already there.
Yeah. Oh, that's cool.
So my first question is about the role of the demyelination versus axonal degeneration. Also, I'm wondering how specific MRI is to those. I feel always like a DTI or MWF or like a mechanization transfer based, or they are sensitive both.
Absolutely, yeah. So we correlated the MTR to because myelin water fraction, we are really trying to isolate the fraction of the water, and also we validated using omics, which was really a good specificity using transcriptomics and proteomics. So right now it's the gold standard for quantifying myelin content. So we correlated with MTR, but we saw a moderate correlation with myelin water fraction. And this is expected because, you know, the signal there is really an average across the different water compartments. So it's not separating really the water contribution from each water, the signal contribution from each water compartment. So it's an aggregate measure average over, it's like T1 or T2. But another interesting aspect is that you can use quantitative MT where you acquire data with different off-resonance excitations and TR, and then you can do a multi-component system to do your inverse problem and isolate the fraction of the lipids from the fraction of the extracellular water. But again, it's a scan intensive, so you need a large amount of data to be plus, not really practical, at least in our investigations here.
So are we all able to differentiate myelination versus external damage? Like a combined maybe myelin plus ETI approach?
That's what we wanted to do that. The issue is that why we combine multi-component relaxometry and multi-component diffusion. There are physiological aspects and experimental aspects. The experimental aspect is that when you use diffusion like a DTI and you do like an API scanning, right? Your echo time is very, very long because you have all your gradients to go cover your case space. And that increases the echo time. So you are no longer sensitive to the signal from the myelin, which is around like 15 milliseconds. When your echo time is 70 milliseconds, you lose all the signal. So there is no contribution for the signal from the myelin, right? And also, if you want to quantify myelin water, which is diffusing really slowly, you need very, very strong gradients, and we don't have that to do that, even if you shorten your echo time. So you need to combine them, measure them separately. On the other hand, for relaxometry, you can differentiate the T1 and T2 from the myelin, but the intra and extracellular water exhibit similar relaxation time, so you cannot tease them apart. So you need to use multi-component relaxometry to quantify myelin independently from multi-component diffusion to quantify the axonal density. Theoretically, what you are seeing is feasible, but on paper, but not on like a practical way due to hardware limitations and physical aspects.
Yeah, another question is about the regional. Mentioned that I'm wondering, you have a lot of data of the results based on the average.
Yes.
Yeah, how about regional myelin?
That's a fantastic question. So what we showed here with all the superiorities of diffusion as compared to PET and CSF, all of that, as you said correctly, is based on an average value across all white matter regions. But we start looking at this, and we can even improve this predictive power and discriminatory power when you look at a specific brain region, especially the anterior brain regions. Frontal lobe and parietal lobe, you can see a much earlier damage. And this is expected because those are the brain that maturates really early in the brain maturation and development, and consequently, they are very sensitive and to brain insults, so external insults, etc. So yes, it will be useful to look at, and we are doing that. And also correlating specific brain regions to some cognitive measurements, like executive function, processing speed, more specific to any changes in specific cognitive.
Based on the data you've seen so far, do you think that the axonal degradation and the demyelination occur independently, or do you think one might influence the other, because that could, open the door to which treatment direction?
You guys ask only fantastic questions. Yes, that's a question that we are looking at. We would like to look at the temporalities, because there are different scenarios. As I said to Dr. Ragh, if it is determined by some genetic factor, they might happen at the same time. But also, if they are not, probably you would imagine that demyelination will be first, and then the axonal degeneration. So we are looking at some statistical ways to look at the temporality and also the correlation of those physiological aspects, myelin axonal degeneration, and also looking at different brain regions with different myelin aspects and different axonal density to see the general help me.
Generalizability.
That's fine. Very good question.
I was wondering if you're able to ask the question present for the differential pattern.
Yeah, so we haven't looked at vascular dementia, but we are hoping to collaborate with another to look at frontotemporal dementia. as we suspect, like at the frontal regions, or at least, which are our hypothesis is that those regions maturate very late in the progression and maybe must be vulnerable, which may lead to frontotemporal dementia. But for the vascular dementia, we haven't looked at that, but we would like to have access to the data there. What is this disease that has a vascular origin? Cassidil?
Oh, yeah.
Yeah. You don't get your lunch. Hungry. So that would be a very good model to study this aspect. Unfortunately.
I'll ask one more. This is too hard of a question, but you left too much time for questions. So given that a lot of this, maybe not, could be Wallerian degeneration, do you have any thoughts on how you can put like some gray matter sequences with white matter sequences in a way that we can start kind of chipping at that question?
Absolutely. So that's a very good question, and we start doing that. Like my postdoc Jonghyun Bae, he submitted an K99 grant looking at the gray matter. The problem with gray matter is that it's a little bit tricky, because if you want to quantify myelin in the gray matter, you really have to take account of the exchange. Because if the amount of myelin, there are two aspects. Like the myelin is very low in gray matter, so you need really some good SNR to have like a quantification. And also you need to include the exchange, because the water exchange becomes much more higher, which adds complexity in the inverse problem. So we're trying to find a way around that using machine learning and deep learning, training some models, but we need some gold standard data. And with our 7 Tesla, we are exactly looking at that. For axonal, or in this case, a neurite degeneration, because we are talking about gray matter. So there are some sequences that can account for this exchange, such as the NEXC model. But you need, again, to acquire data with different echo times, different p-values, extended acquisition time. And we are trying to find ways how we can accelerate. And I think this question where machine learning and deep learning may be very technically helpful tools, because we are really reaching a limit here about acquisition time that you can do as much as you can.
Well, thank you very, very much. That's a great talk.